Hello, my name is David Langston. I'm a plant pathologist located at the Suffolk Tidewater AREC Research and Extension Center uh, with Virginia Tech. Um, Today I'm going to talk about a new corn disease that we've been noticing in the southern and mid-sized states. Um, I would like to acknowledge Dr. Michele Batista. She's a postdoc that's been working with me on this project since we began. So this disease was reported first. Actually, it was reported, but wasn't reported as a disease on Crop Protection Network back on in July 2021. You show the pictures here. Give photo credit to Kirsten Wise at University of Kentucky. But people were seeing this, mostly extension specialists were seeing this in their corn trials and in some grower fields. It didn't really fit a lot of the descriptions of the diseases that we're used to seeing on corn. You could see some pictures here. You'll notice some great leaf spot lesions, the very long uh, lesions, but then the tiny lesions. It's hard to pick out individual lesions that coalesce. Um, is the new disease that we're talking about it. Um, but it actually it showed up in once again it was first reported in 2021. No pathogen. That report said that no pathogen had been associated with the symptomatic leaves. Nobody had isolated a pathogen, and in that particular article, it had been classified as an abiotic disorder. And in talking with other folks, that's what that's what we're calling it. And each year, the symptoms appeared after tasseling and into grain field, and it tended to develop in the heat of the summer. The severity of the symptoms were different across hybrids, as people described. Nobody had reported it in any yield loss. And I actually observed it in my trials in July of 2021 as well, which was a big help to me because I didn't know what it was at the time. So it was very serendipitous that this article came out. And so in 2022, we detected it in field trials. We were seeing differences in fungicide treatments. Um, we we started rating it, and eventually we isolated from infested leaves, and every isolation that we isolated was a what we're calling diporthay soj. So we thought we knew what it was. And in one sample that we looked at in 2022, we actually isolated a Negrospora species. So going into a description of this disease, the disease typically in my plot shows up suddenly at R2 or blister stage, and it typically shows up on the leaf just below the ear leaf and upwards. We typically see it worse on field edges, more so than we do in the field. It's more prevalent in corn planted in wider rows, 36-inch rows versus 30-inch. And in my area, most of the corn is planted on 36-inch rows versus 30. Lesions are tiny. Most of them are less than a millimeter, so they're much smaller than most lesions that we see with other diseases. These tiny lesions will coalesce and they form longer, elongated necrotic areas. Large areas uh, can of affected leaves take on a peach or a rusty color. So these are some close-up shots, and the disease that I would confuse it with most would be curvularia. So on the left, we're looking at the new corn disease, showing how tiny those lesions are and how they coalesce and they're clumped together, so to speak. When we look at curvularia from pictures taken from CPN, um, we notice that they're individual spots they don't coalesce. Some of the spots are very round, and they're bigger than those that we see with the new disease. So lab observations in 22 were that we collected some leaf samples, we placed them in a humidity chamber for 24 hours, and when I looked at them, I had just looked at gummy stem blight on watermelons, which the asexual stage is a phoma, which is also related to phomopsis. And I noticed that the alpha conidia looked a lot like what I was seeing on that gummy stem sample. So I felt like it was close to it was either a phoma or a phomopsis um, at that point. And we do see pycnidia from time to time. So if you see in this picture, if you look in the very center of it, that's a pycnidium of a fungus that's very similar to a pycnidium that you would see with phoma or phomopsis. When we dissect these, cut apart these pycnidia, we see numerous alpha conidia in those. This is dissected directly from leaf tissue, so that everything's kind of pointing towards a phoma or phomopsis, a fungal pathogen. When the season is over, I went out and looked at some senesced leaves that were on the ground behind the corn picker. And you notice these black areas here in that senesced tissue. These are really tiny pycnidia. Um, and they can be a millimeter or more than a millimeter, but they're very tiny. And there's not a lot of pathogens or fungi that cause lesions on corn that produce these type of fungal fruiting bodies. So we're still leaning more towards a phoma or phomopsis. So we isolated on PDA. I said before that mostly everything that we would isolate looked like a phomopsis that we'd see on soybean. Um, and so I dissected some of the stroma. You see that in the middle picture, spore mass that's oozing out of that. And on the right, the last picture, D. Now, this is a picture taken out of Journal of Phytopathology, but it's very similar to what I saw. So I'm using it as a example. But these are beta conidia in image D on the end. And once you see those beta conidia, it's almost like a confirmation of phomopsis, which is really odd because when we think about phomopsis, and I see it a lot on soybeans, and the cultures look identical. We don't think of phomopsis as a disease, as a pathogen on basically monocots. It's very rare on monocots. It certainly would be extremely rare on corn, and there's been no phomopsis disease ever described on corn to this date. So we were scratching our heads on this one because it doesn't seem to fit real well. Dr. Batista sequenced um, the the TEF section of the DNA and ran a blast search, and it seemed to group best with diaporthase OJ. So everything's kind of fallen neatly into place, even though it still doesn't fit with what we normally see with this particular type of pathogen. The frustrating thing is to be able to prove Koch's postulate. So we can't name a disease or name a pathogen that causes a disease until we run this Koch's postulates, was basically a proof of pathogenicity test. So we had completed the first part, which is pretty easy. We saw the disease consistently in different cornfields. We isolated the pathogens, and mostly what we isolated, as I showed before, on the top row of pictures was a diaporthe that molecular identification pointed towards diaporthase OJ, but we also would isolate a good bit on Nigrospora species. So we're not 100% sure with the frequency that we isolate Nigrospora if it's that pathogen or diaporthe. Our biggest problem is that we can't complete Koch's postulates because we can't reproduce the symptoms on corn. We've conducted multiple greenhouse studies on corn and have not been able to reproduce the symptoms. So we can't really name it yet. We're still working on it. We've been frustrated with this disease. It's a hard act. It's a difficult actor to work with. Moving on to some of our data and field trials, 2022. Once again, we saw it. We saw the disease. Um, we noticed that there was differences in fungicide treatment, so we went out and rated this this thing, um, and we evaluated disease on August the 8th. The fungicide data on the second rating, which was on August 25th, showed a good activity with different fungicides. Untreated is the blue bar on the left, and basically every fungicide tested was, reduced disease compared to the untreated in this particular trial. So we're almost everything's kind of leading towards a fungal disease since we can have good activity on it with a fungicide application. And so we conducted fungicide efficacy trials again in 2023. We had stark differences in disease that year. If you notice on the right, the untreated plots, you can see some symptoms, discoloration throughout the plot. And if you look at a treated plot, the best treatment is actually Veltima. Basically, Veltima almost eliminates this particular disease. So it's stark differences between what you see with fungicides compared to untreated. Here's some data from that trial. The green bars are the earlier rating on July 27th. The brown bars are ratings taken from on August 10th. And so we've gotten to untreated all the way over on the left. Um, we got significantly more disease in the untreated than all the fungicides except trivapro. And we've seen this before with trivapro with this disease, that we'll actually sometimes see a little bit more disease in the trivapro treated plots than the untreated, which we can't really explain that. But the trivapro typically runs along just the same as the untreated. We see big differences, significant differences in disease control with all the other treatments. One thing I'd like to point out is that if you notice this Veltima treatment here, this treatment was applied 14 days after tasselin. And you notice how the rating on July 27th is equal to that taken on August the 10th. And you see how it compares to the VTR1 application of Veltum at the same rate. Basically, no difference in disease between the early application or even the later application, almost identical amount of disease in each treatment, which something we never see with fungicide treatments typically is a fungicide actually stopped it, which shows the active ingredients in Veltima must have a curative mechanism of activity against the pathogen. Once again, I just feel like I need to point it out because this is something I've really never seen in a field trial over 25 or 30 years. When we look at the yield from that trial, and these are small plot replicated trials, so it's hard to get yield differences in corn treatments with small plot trials. You notice the untreated control is all the way on the left, and the red bars shows that all the way across all the treatments. Really, the only treatments that I yielded that numerically were Stratego, Absolute Max, and Trivapro. So it doesn't really correlate well with disease. The amount of disease you see doesn't necessarily correlate with yield. And these, it says only really a 10 bushel difference between the untreated control and Trivapro, and those differences were not significant. So in 2024, I submitted a proposal to of Virginia corn board, and these are I tried to trying to get differences or observe differences between treated and untreated. I conducted some strip trials, larger plots. These are strip trials that were replicated 4 times with and without the Veltima treatment. This is a disease severity rating taken on August the 7th at location 1 and location 2. So there's 2 strip trials. We see big differences in the treated and untreated here. The untreated the blue bars showing significantly higher foliar disease levels than we see with the orange bars, which is the Veltima treatments over both locations. But once again, in those same strip trials, we were really unable to record a significant yield difference or even some type of trend in a yield difference. At location 1, we did see a little numerical bump in yield, but in location 2, it was basically reversed. So we're still unable to, even in large trials, that year we were unable to see differences in treatments. Also in 2024, I rated some corn OVT trials or corn variety trials that our corn agronomist conducts every year. And I didn't want to get into the weeds on all the different varieties. The main thing I want to show you is there's a big difference in how varieties react to this pathogen. So there's several, as you can see, several varieties in here. The variety that had the most disease had over 60% leaf area affected on the ear leaf, compared to the least susceptible variety on the far right, having just over 10%. So we do see big differences here, big significant differences between varieties. Here's just another location up in Mount Holly, Virginia, probably about 4 hours away north of us. We see basically the same trend, big significant differences between varieties, a little over like 36% disease in the worst variety, and actually about 1% disease in the least susceptible variety. So big differences. But I couldn't find a common a variety between these 2 trials that correlate well with disease. So I wasn't really able to say when I looked at this variety data that it was a certain corn varieties in general were more susceptible. It was kind of all over the place. And so in 2024, we also we surveyed the disease and took disease samples, and the geographic this just shows the distribution of it that year. I suspect I have actually observed this disease a little further west than the Sperac location. So I do know that it does occur a little bit further west, but even further west of us in Kentucky, we got samples from Dr. Kirsten Wise, and we isolated actually the Nigrospora pathogen from it, but it was the same basic disease symptoms that we see here, where we isolate mostly Phomopsis, but we also isolate Nigrospora. So based on all the data that we'd accumulated since 2021, I was asked to modify the original article where we actually call these symptoms that were reported in 2021 are actually caused by a new disease that is yet to be identified. So it's no longer associated with some type of abiotic disorder or stress symptom or a spray burn. Now when we observe this, we can say that it's caused by a pathogen, more likely a fungal pathogen. So in summary, we see this problem every year, at least in Virginia. I talk to people in other states and they see it as well. I talk to people further south, and especially in Florida and Georgia, And some seed reps down there, they see it a good bit to different degrees of severity, and they agree that they see it more in some varieties than others, and that they also see less of it in fungicide treated trials compared to untreated trials. Last year in 2025, they had a widespread outbreak of it. I got a call from a pathologist at the University of Georgia and was sent pictures of it, and it was like all at once, everybody was seeing it everywhere. And I made the comment before that this disease comes on very suddenly around R2 growth stage. So it seems to be driven by a physiological change in the plant that occurs when you shift from vegetative to reproductive phase. Typically, fungicides are very effective. The most effective fungicide that we've observed is the Veltima fungicide. Kind of getting a little repetitive here, but we also, we see varieties that are more susceptible than others. Haven't seen any yield loss up to this point. Not much different than we see with a lot of other corn disease, especially in small plot trials. But it's really the only disease that I see in my trials. If it wasn't for the new disease, I wouldn't have a whole lot of disease to rate in my fungicide trials, at least in recent years. And we're still trying to identify it. We're trying to come up with new ways to try to prove pathogenicity. We're actually inoculating both Nigrospora and the diporthay fungus to try to reproduce symptoms, so we can go forward with trying to actually name it as a disease of corn. Thank you for your attention. My hope is that people can recognize it as a disease and not some type of physiological injury or spray burn. And to me, if that's that right now, it's more of a clinical or diagnostic of clinical and diagnostic importance compared to something that's going to cause a lot of yield loss. Thank you.